Hey, what's up? How's it going?
Hey, I'm good. How are you?
I've been good. How's your summer been?
Pretty good.
Awesome.
Pretty good. Yeah.
How's the studying been going?
It's been going all right. I'm kind of behind schedule, but
Uh,
I've been studying a bit every day, so I've been making that effort.
that's great.
So, I just had a question. So, I'm taking my MCAT in January.
Yeah.
So, how do you recommend like scheduling out my studying until then?
So, have you like reviewed the content for the exam?
I'm currently like reviewing content right now.
Hmm.
I'm doing chemistry right now.
So I think a great way you can review content, but still get practice is do practice questions. So, if you do UWorld, they have a really great question bank that covers a lot of the content, but you get to actually apply it. So it's
Okay.
pretty it's a better way to learn it rather than just reading the book. So if you could look into UWorld, I think that would be pretty helpful.
Oh yeah, I'll look into it. Thanks.
Yeah. Um, yeah, but probably doing as many questions as possible. Would be a more efficient way to learn the content.
Okay.
And
Do
that
you
mm.
Oh, sorry, go ahead.
Oh yeah, and then that way when you take the real test or do the AMC test, the questions won't feel as hard because the UWorld questions are slightly harder.
Okay,
Mm.
I see.
Yeah. So wait a few more minutes. So let's wait a few more minutes before you get started. Okay. Go ahead and get started. and anyone who joins, we can just fill them in. Okay, so uh, today we're going to be doing a biology passage from the MCAT. And so this passage is called COVID-19 and RAAS, so like RAAS. So before we get started, usually we do like a quick icebreaker. So you could just say your name, what school you're from, and then today's what was the most fun you had over the summer. So
All right, I guess I'll go first. My name is Ashrit, and I went to VCU.
hmm,
And the most fun I had over the summer was going to the beach with my family.
yeah, nice.
Mhm.
What beach did you go to?
We went to Outer Banks.
Oh, nice.
Yeah.
Hi, my name's Treaty. I go to VCU, and the most fun I had over the summer was a night out with my sister in Virginia Beach.
Oh, nice. My roommate, he used to live in Virginia Beach, or actually, he still lives in Virginia Beach, but he was from there.
Yeah, it's a nice area.
Yeah. So my name is Siddharth, but I go by Sid, and I graduated VCU last semester. And the most fun I had over the summer is I went to Rolling Loud in Miami, which is like a hip hop festival. So that was pretty fun. So all right. So like we said, today is a biology passage. So we're gonna go through some of the important concepts to know before we actually look at the passage. So I guess first we can start with what a transmembrane protein is. So one of the biggest macromolecules in people's bodies are proteins. And so a certain type of protein is a transmembrane protein, meaning it runs through the cell membrane. So part of the protein is going to go through the cell membrane. All right. And so proteins are made up of amino acids, amino acid residues, and they're all linked to each other. So linked to each other in a chain. So that's just a basic concept to know. And then proteins have different regions. So each part of a protein is responsible for a different thing. And those are called protein domains. All right. Does anyone have any questions about that? All right.
No.
All right. And then an important thing to know for the MCAT is how your body regulates blood pressure. So basically, the amount of fluid volume, meaning like how much volume of blood is in your body, that kind of dictates what your blood pressure is going to be. So if your fluid volume is high, you're going to have a higher blood pressure because you can just imagine more blood in your body banging against like the vessels of the wall. That's going to create more pressure. And then if you have lower fluid volume in your body, you're going to have a lower blood pressure. Uh, so that's kind of how blood volume relates to blood pressure. And so your body has a lot of steps to regulate it to keep your blood pressure constant. And so you don't really need to know the exact details of this, but if you have like a general idea of how it works, it'll be pretty helpful. So basically, so if your body senses a drop in blood pressure, can you guys see my mouse?
Yeah, we can see it.
Okay. So if you have a drop in blood pressure, uh, it's usually going to mean a drop in fluid volume too. So your kidney is going to release what is called renin. So renin is a hormone, uh, and it's going to go and it's going to cleave this molecule called angiotensinogen. That's going to turn to ANG1. Um, then your lungs are going to release this enzyme called ACE. ACE is going to cleave angiotensin 1 to angiotensin 2. And so the real molecule that actually has an effect is angiotensin 2. So it's going to go to your adrenal gland, which is one of your endocrine glands, meaning it just releases hormones, and it's going to stimulate the release of aldosterone. So aldosterone is going to go back to the kidney, and it's going to lead your kidneys to reabsorb salt. So meaning salt is going to go back from your kidneys into your blood, and then water is going to go with it. So your blood volume is going to get higher. And then it also goes to your blood vessels and it makes them constrict. So if your blood vessels constrict, that means you have like a backlog of blood. And so your blood pressure increases because it's just like more blood in a crowded space. So that's how your body can increase the blood pressure when there's a drop in it. Does anyone have any questions about that? It's pretty a lot of steps in that.
So like, would we be given, is this just background knowledge good to know in case like there's a passage that mentions these things?
Yeah, exactly.
Okay, gotcha.
Yeah. So usually they won't ask about like what angiotensin 1 and angiotensin 2 are. That's kind of specific. It could happen, but most of the time you just need to know is that when you have a drop in the blood volume in your body and a drop in blood pressure, there's going to be renin, which is going to lead to angiotensin 2 being produced, which is going to lead to more aldosterone and more sodium reabsorption and more water reabsorption. So it'll just lead your blood pressure to rise. And after this is done, I'll post this PowerPoint video on the website, so you can like look at this diagram in more detail.
Do you share the slides as well?
Yeah, so the PowerPoint is on the
Okay.
website. So
Oh, okay, gotcha. Thanks.
yeah. All right. Okay, and then so we talked about what a protein is. It's just a collection of amino acids, but each amino acid comes in a different type. So we have charged amino acids. They can have like a negative or a positive charge. There's polar amino acids. So does anyone know what polar means?
Yeah, I think like it has two as a positive and then a negative end, and it attracts like other polar molecules.
Yeah. So polar is kind of like a charged molecule, except it's like a partial charge. So it's not really a real charge, but it's like a partial charge basically. So the electrically charged amino acids have a full charge. A polar is just like slightly charged. That makes sense. And then we have hydrophobic amino acids. So hydrophobic, they're not polar, they're not charged. Um, so they have no partial charge or real charge. And so basically how protein interactions work. So proteins are going to bind to each other when they have amino acids in common. So two proteins may bind through their hydrophobic side chains, because you want similar amino acids to be in contact with each other. So you would not want a charged amino acid to be in contact with the hydrophobic side chain because they would it wouldn't be thermodynamically favorable, just because they would like kind of repel each other. So it's kind of like like dissolves like. It's kind of the same idea as that. Like you want polar with polar, non-polar with non-polar. Any questions about that?
Are hydrophobic interactions usually like the strongest one out of the weak forces?
Um, I think I don't know what weak for like. I think the charge are the strongest usually. Um, yeah, I think charge would be the strongest. Then probably then polar, then I would say hydrophobic.
Okay.
Yeah, that's what I would go with. Um, yeah, and so okay, so yeah, any other questions about that? All right. Okay, and so another kind of important concept to know on the MCAT, so they asked a lot of questions about like viruses. So what you need to know is so viruses can be very different. So there's not one sole way that viruses do what they do. There's a lot of differences between viruses. But the general idea you need to know is first, they're going to bind to the outside of the cell. And so you can have a virus who the membrane of the virus fuses with the cell membrane and then it kind of injects itself inside. Or you can have like a cell receptor on the outside. Um, Let me see if I can. So Can you see what I'm drawing right here?
Yes.
Okay. So you could have a cell receptor on the outside, and the virus could bind out here. So that's one way you can enter. Or the virus could just come directly to the cell membrane. And let's say it has a cell membrane on the outside. That. So let's say this like thing I'm drawing around is its cell membrane. The virus cell membrane could fuse with this cell membrane and it could go inside. Or this whole complex, the receptor virus complex, could also go inside. So there's a lot of different ways it can enter. But those are some of them. So first, the virus has to get inside. And once it's inside, the genetic material. So viruses can be RNA or DNA viruses, and that genetic material can go inside the cell. And so once it's inside the cell, if it's an RNA, what usually happens to RNA within cells, like any cell?
Gets like transcribed and translated and degraded afterwards.
Yeah, so it gets translated into proteins. So if a virus has RNA and it injects its RNA into the cell, it's going to use the cell's ribosomes right here to make copies of it. So it'll make copies of the viral proteins, and usually it will have its own. One of those proteins is going to be an RNA polymerase. So the RNA polymerase is going to make copies of that RNA, so the RNA will duplicate. So that's if it's an RNA virus. But if it's a DNA virus, the DNA will just go directly in here, and it'll integrate into the nucleus. So what happens to DNA in human cells? Like, what's kind of the dogma or like process that happens to it? Like, how do you go from DNA to protein?
DNA to RNA to protein.
Yeah. So your DNA is going to get transcribed into RNA, and then RNA will be translated into protein. So if you have a DNA virus, its DNA is going to go here. So this arrow. You're going to have transcription. So transcription just means going from DNA to RNA. And so you'll make the RNA for that virus. The RNA will go here, where it will get translated into proteins. And then you'll get these copies of the virus. Yep. And then let's see. So that's basically how viruses duplicate. They can enter either through their membranes fusing with the cell or binding to a receptor and entering. Once they're inside, they release their genetic material. If it's an RNA virus, the RNA will just go get translated and make proteins, and the RNA will be duplicated. Or if it's a DNA virus, the DNA will be in the cell. It'll be transcribed and duplicated by the cell, and it'll the RNA from the DNA will be turned into proteins. So you'll get copies of the virus. So any questions about that? So this will all make a bit more sense once we see how it applies to the passage. Okay. So another important thing we want to know is what are the type of receptors in the body? So your body has a lot of different sensory receptors. And usually you can tell by the name what type of receptor they are. So like a thermoreceptor is going to measure temperature. An osmoreceptor measures osmolarity. An osmolarity is just a chemical concentration of something. And then a baroreceptor measures pressure. So there are a ton of different receptors in the body that measure different things. So it's just good to be aware of them. A photoreceptor measures light. So that's all you need to know for that. think now we can get to the passage. Does anyone have any questions about the PowerPoint?
Nope.
All right, sounds good. Okay. I'm sharing here. All right. So does someone want to read the first paragraph of this passage?
I can read it. Is it? I'm sorry if I butcher up certain things.
No, that's fine.
Angiotensin converting enzyme 2, ACE2, is a transmembrane protein composed of 805 amino acids. It is found attached to cell membranes within the intestines, lungs, kidneys, testes, gallbladder, and heart, as well as in a soluble form. ACE2 acts as the SARS-CoV-2 virus receptor. SARS-CoV-2 is a member of a family of viruses called coronaviruses. In addition to acting as a receptor, ACE2 also heterodimerizes
Yep.
with the neutral amino acid transporter neutral amino acid transporter 1 and regulates its functions in gut epithelia. ACE2 is a significant controller of the renin angiotensin aldosterone system, which affects blood pressure, volume, and electrolytes. The COVID-19 agent SARS-CoV-2 binds to ACE2.
All right, nice. All right, so let's break down what that means. So what's the main subject of the passage?
They're basically explaining the characteristics of the virus receptor and like what it does.
Yeah, exactly. So the main subject is ACE2, which is a receptor for the COVID virus. And then it tells us here that it's attached to cell membranes. So within these different organs. But there's also soluble form. So there's one that exists within and outside the cell. So you want to remember the cell membrane is hydrophobic. So soluble things won't exist in the cell membrane because they're most likely polar. So that's why we have two forms of membrane form, which is non-polar, and a soluble form, which is polar. And so it tells us it acts as a virus receptor. And it's part of a family. The virus is part of the coronavirus family. And it says it also heterodimerizes with BOAT1, which is an amino acid transporter. So does anyone know what heterodimerize means?
Well, hetero means different, but dimerizes I'm not too familiar with.
So dimerize just means like come together with something else.
Oh, okay.
So like the ACE2 is different from BOAT1, but they both come together.
Okay.
Yep. Let's see. And so it says here that it's a controller of the renin angiotensin aldosterone system, which we talked about. What does that system do? Like, what's the main purpose of that?
It controls blood pressure.
Right, exactly. And so the COVID virus binds to ACE2. And so remember how I talked about how the virus can bind to a receptor, then that complex comes within to the cell. That's kind of what we're looking at right here. So you see the COVID virus binds the ACE2 receptor, and that whole complex together comes inside the cell. Okay. And Asha, do you want to read this paragraph?
Oh yeah, sure. COVID-19 has had a significant global impact compared to the 2002 SARS outbreak due to mutations that changed its binding affinity. ACE2 genetic variations affect its function and structure, thus altering its affinity towards SARS-CoV-2. In SARS-CoV-2, the region that interacts with the metallopeptidase domain of ACE2 is the receptor binding domain, RBD, that resides within the spike S protein. The binding of the RBD with the ACE2 receptor occurs through hydrophobic interactions. These interactions cause structural changes in the S protein to enable binding. And MACE2 is composed of an extracellular transmembrane and cytosolic domains. The extracellular domain contains a zinc metallopeptidase catalytic site that hydrolyzes angiotensin 2, ANG2. The extracellular N-terminal domain also contains an S protein binding site where SARS-CoV-2 binds amino acids 1 to 716. MACE2 consists of a short transmembrane domain, amino acids 741 to 763, and a C-terminal domain facing the cytosol. The enzyme ADAM17, a disintegrin and metallopeptidase domain 17, cleaves ACE2 at amino acid 716 to 741. ADAM17 catalyzes ACE2 ectodomain shedding. This catalyzes the formation of S-ACE2. During SARS-CoV-2 infection, the binding of the ACE2 receptor to the viral S1 protein promotes the cleavage of the ACE2 ectodomain by ADAM17. ACE2 is subsequently internalized with the viral particles into endosomes. ACE2 processing and internalization reduces its expression in the membrane, affecting the regulation of ANG2 and promoting RAAS imbalance.
All right. So that was a lot going on. So let's kind of break it down. So it says that COVID has had a big impact because of mutations that changed its binding affinity. So if it was going to have a bigger impact, would that mean a mutation with greater binding affinity or lesser binding affinity?
Greater?
Right, greater. So if COVID was going to be more impactful, it would need to bind to the receptor more. So you would look for mutations that increase binding affinity. Okay. And then so later on in the paragraph, they're going to talk about what determines the binding affinity. So it says that ACE2 genetic variants. So remember, these are just variants of our receptor ACE2. Affect its function and its structure. So depending on how you mutate the receptor, that's going to affect how it binds the COVID virus. So that's all it's saying there. And it says in the SARS-CoV-2, which is our virus, the region of it that it interacts with the metallopeptidase domain. So remember, what did we say protein domains were?
Different, chemically different regions of a protein.
Yeah, it doesn't have to be chemically different, but just, different regions of a protein. So they're just responsible for different things. So the COVID region that interacts with the metallopeptidase domain of ACE2 is the receptor binding domain. So the RBD of the COVID virus is going to bind to the metallopeptidase domain of the receptor. So ACE2 is the receptor, and the COVID is the virus. So the receptor binding domain that resides within the spike protein. So you might have heard the spike protein of COVID, but so this receptor binding domain of the spike protein of COVID is going to bind to the ACE2 domain. So all this is telling us is what part of the COVID virus is binding to what part of the ACE2 receptor. So the metallopeptidase domain of ACE2 is binding to the RBD of the spike protein of the COVID virus. Okay. Oh, shoot. I didn't know that the Zoom meetings had time limits on them. It's crazy. Okay.
Yeah, I think they give like 50 minutes or an hour.
Yeah. Let me see. If it ends, then I guess we can just leave and come back. But yeah. Okay. So okay, when the RBD binds to ACE2, that causes structural changes in the S protein to let binding occur. So M ACE2, it tells us it has 3 different domains: an extracellular, a transmembrane, and a cytosolic domain. So the extracellular domain contains a zinc metallopeptidase catalytic site. So this is the part of ACE2 that hydrolyzes angiotensin 2. So how it regulates the whole RAAS system, which is the renin aldosterone system, is that ACE2 hydrolyzes ANG2. So, and the part of the protein that's responsible for that is the part that's outside the cell. So imagine the cell membrane's like right here. The protein is going to cut through it. So this is the part that's going to interact with the ANG2. Let's call this A2. So the outside part is going to hydrolyze it. The hydrolyze just means like break down. Okay. And then the extracellular part also has an N-terminal domain, where COVID binds, which is 1 through 716. And then tells us there's a short transmembrane domain, so it's a part that cuts through the membrane. And there's a C-terminal domain, which faces the cytosol, which is the inside of the cell. So the C part is going to face this side of the cell. And so basically, there's going to be this atom 17. So I'll say 17. So this enzyme atom 17 comes and cleaves at 716 to 741. And so this is called ectodomain shedding according to the passage. And that leads to the formation of soluble ACE2. And during SARS-CoV-2 infection, when it binds to the COVID virus, the S part of the COVID virus, the ACE2 ectodomain gets cleaved by atom 17. Let's try to see if I can upgrade really quick. Let's do that. Sorry about that. I didn't know there's a time limit on this.
Okay. We can always just like join back if you want. Or you can send out
Yeah,
a new link even. It doesn't matter.
because I used to do some of my VCU email, and I think it was they had like a free pro account or whatever. So there's
Oh, okay.
yeah. Okay. Let's see. Yeah, and then so basically once the COVID virus binds to ACE2, it gets internalized. So ACE2 goes inside the cell and its expression gets reduced, meaning it's not its DNA isn't converted to protein as much. And so that affects whether it can hydrolyze ANG2, and that can lead to a RAAS imbalance. Okay. Okay. So does anyone have any questions about that paragraph? That's a lot. Okay, well, let's do question 1. So question 1 says, RAAS can be activated when there is a loss of blood volume, such as in hemorrhage or dehydration. Okay, so what did we say RAS kind of responds to? Like the renin, angiotensin aldosterone system. What is it responsible for?
The blood pressure, and it has to do with the kidneys.
Yeah. And what receptor is responsible for blood pressure? So what receptor is responsible for pressure in general?
Barrow.
Yeah, so baro is the right answer. All right. So based on the passage, ACE2 most likely facilitates SARS-CoV-2 infection during viral. So how does ACE2 allow the infection to occur? Like, what is its role in that?
It basically is responsible for the functionality of the infection and, like, I guess the shape.
Yeah, so just I guess which option do you think would correspond most closely to that?
Maybe A.
Yeah, so A would be the correct answer. Because so basically
Okay.
all ACE2 is doing is the shape is important because it needs to bind to this. But so basically all it's doing is it's just binding and taking this in. So attachment and membrane fusion.
Okay. Okay, so if it wasn't like a specific shape or structure, then it wouldn't be able to attach, right? Is that why attachment is one of the key?
Yeah, so
Okay,
yeah.
gotcha.
So it needs to be the right shape to bind.
Okay.
But basically because it's just a cell receptor, its main role is just binding the virus and taking it inside the cell. So that would be attachment and membrane fusion.
Okay, that makes sense.
Yeah. So it'll attach and the membrane will fuse together and bring it inside. And so it's not really important for replication expression because that's after it's already inside. It doesn't really have to do anything with that after. And it's not involved in viral assembly and release or the lysogenic phase. So lysogenic is when the virus integrates with the DNA and then it kind of like cuts itself out, which is it's not really related to this. Um, but the clearest way it's related is just binding it and attaching to it. So that's why it's the best answer. And that's I answered that one. Okay. So as described in the passage, which of the following is not one of the functions performed by ACE2? So let's do like process of elimination. Let's look at A. ACE2 is a receptor. Is ACE2 a receptor?
Yes.
Yep. So we can eliminate that one.
And it's an enzyme too, right?
Yep. And why is it an enzyme?
It hydrolyzes angiotensin.
Exactly. Yeah. So that's why it's an enzyme. So an enzyme basically just carries out an activity, and hydrolysis is one of those activities. So it like breaks down ANG2. So that makes it an enzyme too. So we can eliminate that. So ACE2 is a hormone. Is it a hormone?
I don't think so.
Right. Yeah, I don't think it's a hormone either.
Because it doesn't say anything about it like impacting the bloodstream or
Right.
like
Yeah,
any sympathetic nervous systems or anything like that.
yeah, so that's the answer. So hormones have to travel through the bloodstream, but it doesn't say that this travels through the bloodstream. So this, and we said A was wrong, right? B was wrong. And so is ACE2 a regulator of a transporter?
Yes.
And which transporter does it regulate?
Transporter 1, the boat
Yeah.
1.
Yeah, exactly. So it says here it regulates boat 1 right here. So we know this option is wrong. So we know it's not a hormone, so that's why that's the correct answer. And that's why it's correct. Okay. Question 4. So various ACE2 variants differ in how they affect a population's susceptibility to COVID-19. In American populations, there are 2 predominant variants, RS, and I won't read all those numbers, but T27 to A. So whenever you see these letters on the MCAT, they're referring to amino acids. So does anyone know what the T stands for? So T is this is OH. Yeah, so this is threonine. So this is a side chain. So it has an OH group. So if it has an OH group, it's going to be polar or non-polar?
Polar.
Polar, right? And alanine is just the side chain just looks like this. Just a methyl group. So like imagine this is CH3. So is this going to be non-polar or polar?
Non-polar.
Right, because it's just carbon. So carbon is non-polar, but oxygen bonded to an H is always polar. So what are we going? This is what we're starting with. We're starting with T and we're going to A. So what are we going? We're going from polar to non-polar or non-polar to polar?
Polar to non-polar.
Exactly. Okay, so we got that. And then this right here is F to S. Does anyone know what F is?
Phenylalanine.
Right. And is that non-polar or polar?
It's non-polar.
Right. So phenylalanine kind of is like this. Attached to the carbon is a phenyl group, which is like this. Ignore that. So let's start again. Okay, like this. And this is non-polar. So this is phenylalanine. And what is it? Does anyone know what the S is?
Serine.
Right, serine. Yep. And so serine is wait. Yeah, I'm just gonna make sure. Don't want to give you the wrong thing. Yep, you're right. It's serine. So yeah, so serine is going to look like this. So it's going to have an OH group. So is that going to make it polar or non-polar?
Polar.
Right. All right. So are we going from polar to non-polar or non-polar to polar?
Non-polar to polar.
Right, exactly. Okay, so we got that. Let's erase that. So increased susceptibility to the RS781, so this one variant could be explained by increased. So before we even look at the answer choices, what would cause greater susceptibility to COVID? Just based on the receptor and protein properties, what would you imagine would lead to COVID being worse for someone?
The receptor having like a high binding
Yeah.
affinity to the virus.
Yeah. So basically, if COVID can bind better, then it's going to have a way worse impact on someone. So increases susceptibility to this variant. And so basically, whenever the MCAT introduces like amino acid substitutions, you kind of can tell they're trying to test you on like protein binding and stuff like that. So so you know that the increased susceptibility is going to be due to binding, basically. So for this one, the 781, we're going from what are we going from again?
Polar to non-polar.
Right. And so how does the COVID protein, the S protein, the COVID S protein, the spike protein bind to ACE2?
Yeah, hydrophobic interactions.
So if we're going from polar to non-polar, is that going to increase binding or decrease binding?
Increase it.
Right. And that's going to make COVID more deadly. So that's why the correct answer is binding affinity. Right. But let's look at the other answers too. So expression of ACE2. So expression of ACE2 means how often does the ACE2 DNA get transcribed, which means it turns into RNA, and then how often does it get translated? So the mutation doesn't mention anything about how it affects its expression. So we can rule that out. It doesn't say how it affects the catalytic activity because we don't know how the amino acids of this protein affects its catalytic activity. So we rule that out. And protection from host proteases. It doesn't mention anything about that either. So the most clearest and obvious answer is binding affinity. So basically, what they're just testing you on is they're testing you on how amino acids affect protein binding. So that's why binding affinity is the correct answer. Does that make sense, or any questions?
Yeah, that makes sense.
Good. Okay. Last question. So according to the passage, Atom 17. So what does Atom 17 do again? So let's look. It says the binding of ACE2 to the viral S1 protein promotes the cleavage of the ACE2 domain, ectodomain by Atom 17. So Atom 17 cleaves ACE2 at 716 through 741. So let's go through, and then when that happens, it gets internalized. The receptor gets internalized. So ACE2 gets taken inside the cell. And when it gets taken inside the cell, its expression in the membrane is reduced. And the regulation of ANG2 is tampered with, and there's an imbalance in the RAAS system regulation. So let's look at the answer choices. If Atom, what does Atom 17 do again? So Atom 17 cleaves the ACE2 ectodomain. And then ACE2 goes inside the cell. So if ACE2 goes inside that cell because of Atom 17 cleaving part of it, does that prevent the RAS regulatory function of ACE2? So you can just take a guess, yes or no. It's 50-50. So
No, maybe.
that's actually the wrong answer. It's yes. But so it does because so what did we say Atom 17 does? So Atom 17 cleaves ACE2 at amino acid 716 to 741. So it's kind of chopping off some of its amino acids, right? And so when COVID binds to ACE2, Atom 17 is going to cleave part of ACE2. And so when that happens, ACE2 gets taken inside the cell. And when ACE2 gets taken inside of the cell, it can't hydrolyze angiotensin 2. Remember, it says that the extracellular domain of ACE2 hydrolyzes angiotensin 2. So if ACE2 gets taken inside the cell, it can't hydrolyze angiotensin 2. Therefore, it can't regulate the RAAS system, the RAAS system.
Oh, okay, makes sense.
Yeah. Any other questions about that? For just option A.
No, no questions.
All right. So A is the correct answer there, but let's go through the other options too. So atom 17 cleaves ACE2 where the S protein binds. So where does it say atom 17 cleaves ACE2? It says it right here.
Yeah, 716 to 741. Yeah.
Right. And where does the S protein bind?
Amino acids 1 to 716.
Right. So do those overlap? Well, yeah.
No, yeah.
Yeah. So that's why we eliminate that one, because it doesn't cleave where the S protein binds, because the S protein binds 1 to 716. It's the S protein binding site. But this one cleaves at 716 to 741. So this is a different part of the protein. So we erase that. And does atom 17 increase the host resistance to COVID-19 infection?
No, because it binds it cuts the angiotensin.
Right. Yeah. Yeah, so it doesn't tell us whether it increases the host resistance to COVID-19. So we can kind of eliminate that. And does it decrease the probability of so actually so basically when atom 17 cleaves the ectodomain, the ACE2 moves inside with the virus. It's internalized with the viral particles. So it can't increase the host resistance if atom 17 leads to ACE2 being ACE2 and the virus moving inside. So if those move inside, then the virus is going to be more effective. So it's not going to increase the host resistance. So that's why we eliminate that. And so atom 17, we know, leads to the virus being taken inside with ACE2. So is that going to decrease or increase the probability of COVID infection?
Increase it.
Right. So we eliminate that. And any questions about any of those answer choices? Right. And so we can see this one was the right answer. And so let's see in the chat. Yeah. All right. So yeah, so that was basically the end of the passage. And so any questions about the passage or any questions or background PowerPoint? All right. And that's all for today. Unless you have any questions about the MCAT or anything like that. Let me stop sharing. Any questions or anything? All right. I guess, yeah. And if you have any friends who are interested in MCAT tutoring, or just MCAT resources, if you could just refer them to the site, let them know it's all free, and yeah, that'd be great. So I guess if no one has any questions, I guess have a great day and thanks for coming.
Yeah, thanks for doing this. It was really helpful. Thank you.
Yeah, no problem.
Thank you. Have a good rest of your day. Bye.
Yeah, you too. Have a great day.